Welcome everyone who's joining. We're just going to let everyone join and get started in a moment. All right, we'll get started now. Welcome to MycoTalks, hosted by the MRC Center for Medical Mycology at the University of Exeter. My name is Christina Cuomo from Brown University and the Broad Institute. And today's speakers are Yong-Sun Bahn from Yonsei University in Korea and Wendy van de Sade from Erasmus MC in the Netherlands. MycoTalks are hosted by the committee shown at the bottom of this slide with valuable assistance in running the event from the MRC Center staff. MycoTalks are held at 1600 UK time on the last Thursday of every month, and were started by the MRC Center back in July of 2020 to bring together the medical mycology community to hear about the science during the lack of face-to-face conferences during the COVID-19 pandemic. And MycoTalks attracts an audience of around 300 scientists from around the world, with many more viewing the recorded talks later on YouTube. Apart from the great science, the philosophy of MycoTalks is to promote the diversity of our field in terms of career stage, gender, geographic location, and research field, and the committee is always happy to receive speaker suggestions for our future program. The format is 2 30-minute talks on diverse aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will be seen only by the moderators. And it's important that you state the name of the speaker you wish to address. The chairs will then alternate the questions for speaker and may choose to collect some of the questions that are similar together in addressing them to the speaker. Even if we run out of time, all the questions that are posted to the speakers in the Q&A will be sent to them after the event. The upcoming MycoTalks are listed on the website, and you need to register for each session. Again, note the time of the talks is 1600 UK time, and so that in the spring and the autumn, you need to check how this might be affected by changes in daylight savings time. So there is an upcoming talk next month in July, Christina Hull from University of Wisconsin and Thuy Lee from Duke University. And then there will be a 1-month break in August before the next session season 5 will recommence in September with talks from Julia Kohler and James Cronstad. We also wish to draw your attention to the Medical Mycology Training Seminar Series, which is held on a monthly basis and is a great opportunity for early career scientists to give a talk. So there is information to get in touch or submit an abstract via the link on the bottom of this slide here. More recently, we have launched the MycoClinics as a series of online talks specifically with a clinical focus. And again, you can find the schedule for these on the MRC Center website. So now I'd like to introduce our first speaker, Yong-Sun Bahn. Dr. Bahn is an Underwood Distinguished Professor in Biotechnology at Yonsei University and a Fellow of the American Academy of Microbiology. He earned his PhD from Ohio State University, and Dr. Bahn has made significant contributions to functionally characterizing the complex signaling pathways that govern growth, differentiation, stress responses, and virulence in human fungal pathogens, including Candida albicans, Candida auris, and Cryptococcus neoformans. His research utilizes molecular and genetic approaches to enhance our understanding of fungal pathogenesis mechanisms. In addition to his research, Dr. Bahn has advanced the fields of microbiology and mycology, both in Asia and internationally. He has served as an editor for several high-impact journals and as a reviewer for numerous papers in fungal genetics and biology. He's also played a pivotal role in fostering international academic interactions between Asia and Korean microbiologists and researchers worldwide. And his scientific contributions have been recognized with numerous academic achievement awards. So I welcome him and invite him to begin his talk.
Thank you, Christina, for the nice introductions. Okay, so right, everybody can see this, right? Okay. All right. First of all, I would like to thank the organizing committee for inviting me to this famous MycoTalk seminar series. Today, I'm going to talk about our recent works entitled Genome-wide Functional Analysis of the WD40 proteins in Cryptococcus neoformans. Sorry. So Cryptococcus is one of the major fungal pathogens which can cause the systemic mycosis. It is the Basidiomycetes fungus, which is globally distributed in both natural environment and clinical settings. So the most recent global epidemiological study demonstrated that cryptococcal meningoencephalitis is responsible for more than 140,000 deaths globally every year. So due to this clinical importance, WHO has recently categorized the Cryptococcus neoformans as one of the 4 critical priority fungal pathogens groups. So Cryptococcus neoformans is found in diverse natural environment, and there it generates infectious propagule such as basidiospores and dried yeast, which are subsequently inhaled through the upper respiratory tract and initially infect the lungs, causing fungal pneumonia. Subsequently, Cryptococcus can be disseminated into the bloodstream and eventually can infect the central nervous system by crossing the blood-brain barrier, causing fatal meningoencephalitis. During this infectious process, Cryptococcus neoformans can utilize a variety of the different virulence factors, including the ability to survive at the host physiological body temperature and 2 antiphagocytic virulence factors, including polysaccharide capsules and polyphenol melanin pigment and urease, and also various defense mechanisms against the different host-derived stresses. Nevertheless, the pathobiological signaling pathway modulating these virulence factors still need to be further elucidated. Therefore, in past decades, my lab has been working on characterizing this pathobiological signaling pathway in Cryptococcus neoformans and identifying the key signaling component which could be potentially exploited as a novel anti-cryptococcal or antifungal drug target. To elucidate the pathobiological signaling pathway in this pathogen, we have mainly focused on the transcription factors and kinases and phosphatases, which are shown to be key signaling component in most eukaryotic organisms. By focusing on this central signaling component, we believe that we can at least have some general overview of the fungal pathobiological signaling pathway. So this is the schematic for our approach to systematically analyze the functions of these signaling components in Cryptococcus neoformans. For all these 500 signaling components, we first aim to generate the signature tag gene deletion mutant with at least more than 2 independent strains for each gene. And for the constructed mutant, we performed a variety of the phenotypic analysis under 30 distinct in vitro conditions, including growth at varying temperatures and mating and virulence factor productions and various stress responses and antifungal drug resistance. Most critically, we performed the 2 large-scale virulence assay using insect killing models and murine-based signature tank mutagenesis because the conventional animal study will not be feasible for this large set of the mutant collections. So we expect to elucidate the pathogenicity-related signaling pathway. So we have spent almost 12 years accomplishing these 3 projects by constructing a total of 816 signature tag gene deletion mutant representing 155 transcription factors and 129 kinases and 114 phosphatases, and systematically analyzing their in vitro and in vivo functions. As a result, we discovered about total 139 pathogenicity-related TF and kinases and phosphatases. We believe that some of these pathogenicity-related signaling components could be excellent antifungal drug target in the future research. So this is the rough draft for the integrated fungal pathogenicity-related signaling pathway networks. So we found a variety of the pathobiological features such as a cell cycle, regulation, DNA damage response, repair, stress responses, and protein folding and trafficking are involved in the pathogenicity of the Cryptococcus neoformans. We may have to put more effort into improving the resolutions of this complex pathogenicity-related signaling network in the future studies. As Cryptococcus neoformans is the major etiological agent for the fungal meningoencephalitis, so we have been curious about also which signaling pathways are involved in the BBB crossing of this fungus. To address these questions, we utilized our TF and kinase mutant collections and employed the dual STM analysis in the lung and brain. In our previous study, we did the STM analysis of the lung, nasally infected with the Cryptococcus neoformans, TF and kinase mutant collections, which cover mostly the early stage of the infection. To elucidate the signaling pathway involved in the late stage of the infection, we additionally performed the second round of the STM analysis of the brain tissues in the intravenously infected with the same mutant collections, which can bypass the lung infections and thereby examine brain infection more accurately. Through this STM analysis data, we have identified the several kinases and transcription factor involved in the brain infection. Subsequently, using the transwall system containing the monolayer of the human cerebrovascular endothelial cell, we identify the several kinases and TF involved in the BBB crossing of the Cryptococcus neoformans. If I summarize the data of this result, we found that a very distinct but also redundant set of the complex signaling pathways are involved in the whole infection stage of the Cryptococcus neoformans, such as the lung infections and bloodstream dissemination and BBB adhesion and BBB crossing, and there's survival in the brain tissue. One of the major issues in this study is that we used the 2-dimensional BBB transwall system. However, the BBB is not a simple system like that. So, in fact, it consists of the vascular cells, including endothelial cells and pericytes, surrounded by the astrocyte and microglia and the neurons. Therefore, it's very difficult to make an in vitro system simulating real BBB structures and environment. To overcome these limitations, we collaborated with Professor Sumu Jo in our department. We developed the microfluidic system, microfluidic human neurovascular unit chip to simulate the real BBB structures. Therefore, we've applied this BBB chip to investigate the brain infection mechanism of the Cryptococcus neoformans. So once one day after inoculated into the upper channel of the BBB chip, and then as you can see, the Cryptococcus cells can form the cluster near the BBB junction, and then some of them observed to cross the BBB barrier of the BBB chip. So we further investigated Cryptococcus neoformans neurotropism in this multi-organ chip prepared by the adding additional liver mimetic unit into the chip. So in here, by flowing the medium from the right to the left, the fungal cell contacted the liver unit first. So to quantitatively measure the neurotropic movement of the fungal cell, we define the degree of the neurotropism according to the following equations. So in this multi-organ chip, so as you can see, the Candida glabrata was evenly distributed in the whole endothelial cell layer, regardless of the presence of the brain and liver unit. In contrast, the Cryptococcus neoformans cells were more preferentially stacked on the endothelial cell layer above the brain unit. So then the liver unit, showing the clear the neurotropism. Notably, as you can see, the Cryptococcus NPR1 mutant showed the degree of the neurotropism similar to the wild type, but the significantly decreased the BBB penetration, indicating that the metalloprotease NPR1 is critical in BBB penetration of the Cryptococcus neoformans, but not in its neurotropism. Finally, we applied this multi-organ chip to simultaneously screen the novel genes required for BBB penetration and neurotropism in Cryptococcus neoformans. As you can see, that we found that the TRM7, DOC101, and TRK1 kinases and PK1 kinases mutant and the single STIF4 mutants are indeed defective in the BBB crossing. Notably, we found the STIF4, DOC101, and PK1 are also involved in the neurotropic movement of the Cryptococcus neoformans. So collectively, this multi-organ chip could be utilized in future for simultaneous screening of the BBB penetration and the neurotropic movement factors in the Cryptococcus neoformans. So all this the published work. So from now on, I'm going to talk about today's main topic. I thank our collaborators and the funding agency. So if you have any question, I'm happy to answer it. Thank you.
Thank you very much for a wonderful talk. We'll save the questions, as you know, for the end of the session. So just to remind everyone attending that if you have questions, please put them into the Q&A box at the bottom of your screens, and please give the name of the speaker to whom you wish the question to attend. And so we'll come back to questions for Sande at the end. Thank you once again.
All right.
Next up, we have our second speaker, which is, or who is, Professor Wendy van der Sande. So she's an associate professor of medical mycology at Erasmus MC in the Netherlands. Wendy has had a career studying mycetoma even before she completed her PhD, and has made significant contributions to this area. She and her team has developed several diagnostic tools to identify the causative agent and species levels, and these tools have been instrumental and used worldwide. She also leads the bacterial screening program of the MycetOS open source drug discovery prize program, MycetOS. I hope I pronounced that correctly, and that's the title of her talk, so we'll hear more about that shortly. She's also had a lot of community engagement in helping to promote mycetoma as a disease entity. For example, she's the founding member of the Global Mycetoma Working Group, convener of the mycetoma Working Group at ISHAM, the International Society for Human and Animal Mycoses, and is also involved as vice chair of the skin entity subgroup of the Diagnostic Technical Advisory Group, it's a mouthful, at the World Health Organization. So Wendy, we look forward to hearing about your open source drug discovery platform. Thank you very much, and over to you.
Thank you very much. I will share my screen. So first of all, thank you for the organizing for giving me the opportunity to present our open source drug discovery project, MycetOS. And the aim of this project is actually to identify drugs for mycetoma. So for those of you who are not that familiar with mycetoma, mycetoma is a chronic subcutaneous disease which is characterized by the discharge of grains and purulent material through sinuses. Since this is a big mouthful, I will walk you through the process. So in endemic regions, people usually walk barefoot, and most of the causative agents are thought to reside in the soil. So when you walk barefoot and there's a lot of thorns and other things which can penetrate your skin, there's also a risk that the causative agents will be penetrated in the subcutaneous tissue. So in the case of mycetoma, the fungi or bacteria are introduced into the subcutaneous tissue by a thorn prick or another small trauma. And when they are in the subcutaneous tissue, they will have to defend themselves against the host immune system. And they do that by forming typical grains, which you can see in this case as black spots over here. This doesn't hurt, and the lesion often heals, but the fungus or the bacteria is inside and will start to grow further into the subcutaneous tissue, and in the end will even invade the bone, as you can see here, where it forms cavities. So mycetoma is a so-called neglected tropical disease, and it was recognized as such by the WHO in 2016. So most cases for mycetoma are seen in Mexico, Sudan, and India. So since mycetoma is thought to be an implementation mycosis, it's not surprising that most of the cases can be found in the feet. However, there are also numerous cases in the legs and in the hands. And the cases can be as small as this lesion over here, but can also be very extensive. So at the moment, mycetoma can be caused by more than 19 different causative agents, and this number is still growing. But if we look through the 27,000 cases reported in literature, we see that there are a few causative agents which are more common than the others. So on the bacterial side, you can see that most common bacterial causative agents is Nocardia brasiliensis. And on the fungal side, this is the fungus Moduella mycetomatus. You also see different colors over here. So the grains I mentioned before, they can be of different colors. And in the case of Nocardia brasiliensis, they are often whitish, and Streptomyces somaliensis forms yellow grains, while Actinomadura causes red grains. For the fungi, most of the causative agents form either black grains or white grains. So you can imagine if this disease can be caused by bacteria and fungi, the treatment is also different. So in the case of bacterial mycetoma, usually the disease is treated by 5-week cycles of trimethoprim sulfamethoxazole, which is given throughout the full 5 weeks, and amikacin, which is given only the first 3 weeks. And this results in a 95% cure rate. Doesn't mean that this is only a 5-week treatment. This cycle is repeated over and over again till the lesion disappears. In the case of the fungi, the treatment consists of 6 months of itraconazole therapy, then the lesion is surgically removed, and another 6 months of therapy. So the bacterial treatment is very successful, 95% cure rate, but for the fungal disease, this is unfortunately less successful. So in the past, there were there's only recently one clinical trial, but if we look at the case series presented over here, you see that in the beginning days, ketoconazole was the drug of choice and the corrected cure rate was 30 to 56%. But when itraconazole was introduced, it was only 9% if surgery was not included, but it could be up to 100% with surgery. Most case studies show that roughly 60% cure rate is obtained. But this is a cure rate with surgery included. So that means that the drug by itself doesn't do that much. So why is this treatment so difficult? Well, here you can see a mycetoma foot. And when we go inside, you can see all these black grains inside. And if we zoom further into the black grains, you can actually see that the black grain is surrounded by a large zone of neutrophils and other immune cells. And surrounding that, there's a capsule. So inside these grains, the fungal hyphae are even embedded in a cement material. So for a drug needs to penetrate first the capsule, then the immune zone, then it needs to penetrate inside the grain, penetrate all the way through the cement material to enter the cell. So that's a lot for a drug. And in the cell, it's probably also needed to kill. So there's a desperate need to identify a novel treatment for mycetoma. So there are two routes which can be followed. First is repurposing of antifungal agents developed for other fungal diseases. Secondly, we could identify novel drugs by an open source drug discovery approach, and that is called MycetOS. So what is our drug discovery pipeline? So it's a relatively simple drug discovery pipeline. We usually start with in vitro screenings to determine if the drug or the compound can inhibit the fungal growth. Then we use an invertebrate model to see if it can also penetrate the fungal grain. And then the procedure is split. So if we have a very promising compound, we could go into a mouse model and then later into clinics. If the molecule is promising but does show some toxicity, we could modify the molecule and start the in vitro screening cascade again. So what do we determine with in vitro susceptibility? So the first question we want to answer is, can the drug penetrate the fungal cell? And then can it also inhibit the fungal growth? So Margella mycetomatis is a very peculiar fungus. So under normal conditions, it doesn't sporulate. So the normal CLSI or UCAST assays cannot be used. So for that, we developed an alternative where we grow the fungus in RPMI and then disrupt it to get standardized hyphal fragments. But because these hyphal fragments are already turbid, we usually use a viability dye to determine if the fungus still is metabolic active, yes or no. And in this case, we use XCT. So if the fungus is active, the colorless XCT will be transported into a bright orange product. And this can help us with establishing where the minimal inhibitory concentration lies, as you can see over here. So that's the first well where there's no orange seen. So we started by looking at existing antifungal agents, and we looked at the azoles, ravuconazole, itraconazole, posaconazole. We looked at amphotericin B, the echinocandins, and 5-flucitazine. And as you can see, ravuconazole was by far the most active antifungal agent, which had the lowest MIC90 of all drugs. This was then followed by the other azoles and amphotericin B. So from the in vitro results, looking at standard antifungal agents, we could conclude that the azoles can penetrate Margella mycetomatis cells, the azoles can inhibit fungal growth, and ravuconazole was the most potent azole. So then the question arose, can it also penetrate the mycetoma grain? So for that we developed this Scaleria melanella infection model, where we actually inject the hyphal fragments into the last prolag, and then we can treat this same larvae on 3 consecutive days with the dosage of an antifungal agent. So if we treat the larvae with PBS, of course, non-infected larvae will all survive. Infected larvae will roughly all die within 8 days. But during this, the grain is also developed. So as early as 4 hours, you can already see the first signs of grain development. After 24 hours, you can see that cement material is already forming. After 3 days, also immune cells are attracted to the grain. There's a capsule surrounding the grain. And after 7 days, even more immune cells are present. So we can follow this over time. And we start the treatment usually at the time when the grain is already formed. So when we did this with ketoconazole and itraconazole, we noted that a similar survival curve was obtained as with PBS, indicating that itraconazole and ketoconazole treatment alone could not cure mycetoma or penetrate the mycetoma grain. Amphetamine B, on contrast, did prolong larval survival and therefore could penetrate the fungal grain. So this looks contradictory to what we see in patients, because there the azoles are generally used to treat the patients. But when we compare this model to a mouse model we also have, actually similar results were obtained. So our larval model was a lethal model. So amphetamine B was able to prolong larval survival. Our mouse model is a non-lethal model. So in this mouse model, we inject the same inoculum as in the larvae, and you can see grains formed. After 21 days, there are grains present, and often as early as after a week. So when we infect the mice, they are happy. You don't see anything else. But when you open them up after 21 days, they are fully covered with grains. When we treated them with itraconazole, again, grains were seen after 21 days of infection. But when we treated the mice with amphetamine B, no grains were seen anymore. So although the readout is different, the conclusion of both experiments was the same. Amphetamine B did affect the grains, itraconazole did not. So when we looked at the existing antifungal agents, raviconazole was the only azole which could actually prolong larval survival. So raviconazole became also the drug which was tested in the recent finished clinical trial for mycetoma, where fosraviconazole, the prodrug of raviconazole, was used to treat mycetoma patients, and it was compared to the standard treatment with itraconazole. So this was a design where itraconazole and fosraviconazole were given for 6 months. Then in both cases, the lesion was surgically removed. And again, 6 months of itraconazole or fosraviconazole were given. And the results of this clinical trial were just recently accepted for publication, and be on the lookout for that manuscript. But the conclusion was that they did behave similarly in the patients. So there was no benefit of fosraviconazole compared to itraconazole when you look at efficacy. So what now? We need a new drug to treat mycetoma because we want to treat it without the need of surgery. So a few years ago, we stepped into this using an open source drug discovery. And this started by mining compound libraries. So open source drug discovery indicates that it's open source, so everyone can participate. So if you're interested, you can use this QR code to join, to go to our GitHub page and actually see all the results we have gathered so far. So open source drug discovery is a drug discovery project which is conducted in an open forum instead of behind closed doors. So that means that anyone can participate, anyone can help us, but also anyone can see the results we generate at the time we generate them. And there will be no patents. So we started our MycetOS project by the screening of 5 libraries from the Malaria Medicines Venture, which is an open source drug discovery project as well. And this MMV has those compound libraries which you can get free of charge. And each library consists of 160 to 400 structurally diverse compounds. So in total, we screened 1,600 compounds. And here you can see the results. So this is the growth percentage obtained with the in vitro susceptibility assay. So 100% growth means that it's not inhibited in growth at all. And since Margella does form a pigment, we have a cutoff of 20%. So below 20%, we consider that the compound was able to inhibit the growth of Margella mycetomatus. And we usually screen the first libraries at 100 micromolar and 25 micromolar. And those compounds able to inhibit growth at 25 micromolar as well, they are used to establish the concentration where 50% of the cells in a tube are inhibited in growth. So from the 1600 compounds we started with, 404 were able to inhibit growth at 100 micromolar, and 29 of these compounds could inhibit the growth, had an IC50 of 8 micromolar or less. And these were the compounds we were mainly interested in. So what were these compounds? Well, most of these compounds were again azoles. Then we had olorofim, and we had some other compounds. So as you can see, most attacked the normal pathway where terbinafine, the azoles, and olorofim also are, so the ergosterol pathway. But of course, we already knew that the ergosterol pathway wasn't that successful in the treatment of mycetoma. So it's more interesting to look at the other compounds. So olorofim was one of the compounds which prolonged the survival, fenbendazole was one of the compounds, and these MMV compounds. So if we then look at the compounds we found, olorofim did prolong larval survival, and that's an excellent candidate to go further into in vivo models, like the mammalian model, and later on maybe in clinical trials, because it's already quite far along in clinical development. It's currently in clinical trials for other fungal diseases. One of the other compounds was a phenerimol, for which an 800 compound library was available, but the phenerimols are not in clinical trials yet. So this is a very good candidate for this part of the pathway. Because we had this 800 compounds, we could actually mine this compound to allow us to decide which kind of chemical properties are needed to penetrate the mycetoma grain. So for this, we used our open source approach. So what we did was that we selected this compound from one of the MMV boxes. We discussed this on GitHub. And here you see a few of the participants of the MycetOS community. There are biologists, there are chemists, there are even high school students participating. And we discuss each month online about these molecules we found and how we should modify the molecules and test them. When we decide which molecules are of interest, some of them are actually created by the chemists and others are donated by others who already created these type of molecules for other purposes. So for the creation of molecules, of course, we have our chemical partners such as the University College London or the University of Sydney. But more interesting, we also have high school students participating. Like in Australia, they have the Breaking Good project. And in the UK, there are several schools actually participating in this. And how that works is that they actually will have a sort of field trip to the laboratories of our partners. So this one was a school from the Breaking Good project who went to the University of Sydney to make some of the molecules which were shipped to us. And usually the students are very interested and very happy, and they also Twitter about these molecules they make. And some of them have very bright colors, so look very beautiful on pictures. And then in the UK, we have the Whitgift school, which participates, and they even made lab coats with the MycetOS logo on the lab coat. And they also generate molecules for us, and they do that at the University College of London. So from the 800 compound library which was available and the molecules which were made, we had a research question where we wanted to decide which chemical properties were needed to penetrate the mycetoma grain. And for that, we were looking at the molecule. We looked at the different chemical properties, such as the molecular weight, the log-D, so the lipophilicity, the number of rotation bonds, and compared that with the survival curves to be able to draw this conclusion. And in our first paper, we found out that compounds with a low lipophilicity, so a log-D of 2.5 or lower, were more likely to penetrate the mycetoma grain. But this was only based on 5 molecules which were tested in vivo. So we wanted to prove this hypothesis further. So DNDi donated, Drugs for Neglected Diseases Initiative that is, donated 73 extra molecules from the 800 compound library. Based on the structural activity relationships, the University College London made 39, University of Sydney made a few, and students also donated. So we had in the end a library of 159 extra compounds which we screened in vitro, and 25 compounds which were promising enough to test in vivo as well. And based on that, we found 6 compounds which enhanced the larval survival significantly, while the others did not. And this is a good set where you can actually ask your question, what are the properties needed to penetrate the mycetoma grain? So we looked at the molecular weight, we looked at the rotation bonds, and we looked at the log-D. And if you look very closely, you see that at a low log-D, there's a higher survival percentage in the larvae than at a higher log-D. And if we do statistics on this, you can actually see that indeed compounds with a low lipophilicity had a significant were significantly more likely to have in vivo efficacy. So our chemical rule did stand. However, there was one downside on the Fennery moles. They were very active against Mozzarella mycetomatis, but not active against the other causative agents of mycetoma. And in the field, that's not handy because the diagnostics is not very well in place. So our conclusion, 6 out of the 23 compounds prolonged larval survival. There was a trend towards lower log-D and enhanced survival, but the Fennery moles only had activity against Mozzarella mycetomatis. So we then did a different screen where we screened not only against Mozzarella mycetomatis, but we also screened MMV boxes against other causative agents. The second most common one, Falciformis spora senegalensis, Medicopsis romroi, because it's naturally resistant against the azoles, Mozzarella pseudomycetomatis, and Mozzarella tropicana. And from these boxes, 4 were inhibited by all, 4 compounds were able to inhibit all causative agents. And those 4 all belonged to one set of chemical compounds, the benzimidazole carbonates. And these included fenbendazole, carbendazim, and MMV 1782387. So we decided to test those in vivo as well. And luckily, they also were able to prolong the larval survival significantly, as you can see here. So what are these drugs? Well, fenbendazole is a commonly used anti-helminthic drug against gastrointestinal parasites. And in Cryptococcus, also fenbendazole showed good in vivo efficacy in a mouse model. So this seemed to be a very promising series to further continue with. So it became a new series in MycetOS, and we obtained so far 83 additional molecules to determine what actually makes these molecules perform this well. 16 were made by Imperial College students, 30 were obtained from another project from DNDi, 22 had in vitro activity, and 2 prolonged larval survival. And we are still looking for more molecules, and experiments are currently still ongoing. So hopefully we can do a similar analysis as what we did with the Fennery moles. So in conclusion, MycetOS is an open source drug discovery program for mycetoma, and anyone can participate. It uses the same pipeline that led to the discovery of the compounds currently clinically investigated for mycetoma. And it also led to the discovery of compounds with promising activity against multiple mycetoma causative agents. And we have many questions still open and many expertise we need for this project. So if you're interested, please join us, because our envisioned outcome is to identify effective drugs for mycetoma where we no longer need amputations or surgery, and can cure the mycetoma patients. And this is, of course, an open source drug discovery project, so there are many, many people who actually contributed. So thanks to all the MycetOS contributors, and a special thanks to our chemical team led by Matt Todd, the biological team led by me, and the online contributors, of which Anthony and Chris are very active. And of course, all the students, all the high school students who have participated throughout the project and are still participating. So if you would like to help us or join or become inspired by open source drug discovery, please use this QR code to check our GitHub page and to join us. Thank you.
Thank you, Dr. Van de Sande, for this very nice talk and invitation to the community to join your efforts in combating mycetoma. We will now start the Q&A part of the webinar. And if anyone has questions for either of the two speakers, please post them in the Q&A. And if you could list either the name or the initials, that would be a big help. I'd like to start out with a question for Sun. If you could talk a little bit more about your nice neurovascular chip model that you described. I think these kind of systems are just very interesting intermediaries and kind of in C2 systems. And yeah, just a little more about the components. And then kind of when you moved from your endothelial model into that system, what were the differences that you found by moving into this chip model?
So, yeah. So this chip actually has been developed by my fellow colleague in my department, this Professor Seung-Woo Jo. So he is very specialized in the organoids and the microfluidic system. So, I mean, this is, for example, if you look at this, this chip they have like two parts. This part is the blood endothelial cell part, and then here he put the human human neuronal stem cell. So when a stem cell is differentiated, and then they can create the blood-brain barrier between here. So this is a very simple system. So by putting the fungi and by gravity, they putting into here, so they flow from here to this side. So, and then one of the beauty about this is that you can actually monitor the real time, the crossing of the fungus through the BBB. So, and then also this is very nice if you use the multi-organ chip like this, and then there is same blood endothelial cell line and contact with the brain stem cells and the liver stem cells, and then using these multi-organ chips, then you can actually monitor the neurotropic movement. Actually, neurotropic movement of the Cryptococcus neoformans has been proposed by many people, but this is actually the first experiment. They just pass the liver unit and they more preferentially stack up on the brain part. So they clearly have the mechanism that they prefer to to find the BBB part, the brain part. But still, I mean, it could be utilized for the screening for the factors for affecting the BBB crossing and neurotrophic movement. But still, there's a limitation of this system. Still is extremely difficult to make this microfluidic system. So it's very challenging to mass produce this microfluidic system for really high throughput level of the screening. So yeah, that might be a limitation. So yeah.
Did you then maybe more study interactions you first identified in your monolayer system?
Right now we try to so yeah, there's one challenging part is that after we identify this factor is that so we try to do some like transcriptomic analysis and some other omics kind of approach, but challenging part is that it's very difficult to recover the sufficient number of the fungal cells for the omics analysis. So that's why I'm thinking about it. Do we have to do some single cell transcriptomics or single cell kind of thing? So that might be the way to find out, because this is all the transcription factor and kinase. So there must be some effector proteins that really contribute the BBB crossing or neurotrophic movement of that. So it's actually, I didn't focus on that in this part. As you can see that right now we're focusing on characterizing one of the transcription factor called the HABI1, which plays very central role in the whole infection process. So we did some RNA-seq analysis of the HABI1, and then it seems like the HABI1 can control a lot of different effector proteins that some of them might be contributed to the BBB crossing of the Cryptococcus neoformans. So probably in next couple of years that we could provide more detailed data about it. So
Thank you.
Can I jump in?
Yes.
Wendy, I have a couple of questions for you about this really interesting drug discovery platform, but I want to go back to some of the stuff you said in the beginning. You talked about the different presentation of the disease, and you showed some somewhat dramatic pictures of small lesions and large lesions. So I was wondering whether there's any evidence of a difference in host immunity that leads to increased susceptibility, aka worse disease, or decreased susceptibility, and whether anyone knows about that?
Well, the immunology of mycetoma is very understudied. What we see in general in mycetoma patients is that the mycetoma patients have a normal functioning immune system, so they're not immunosuppressed. And with some patients, they have a very tiny lesion even after 30 years, while other patients might have an enormous lesion within half a year. So why some patients have large lesions and others small, it's not a genetic thing of the fungus probably, because there's no evidence that there are differences there. Probably there are differences in the hosts. We found a few SNPs in host genes which were associated with mycetoma development at least. But that's as far as the immunology goes. So there's not much known about why certain patients have enormous lesions and others not.
Okay. And is the pathology, so when you look at the pathology of these lesions, is it different, or does it look the same? So do you see this capsulated, what did you call it, cement-like material in which the oh yeah, they look the same in both?
Yes, it depends on the causative agent. So Margella mycetomatus always has this cement material throughout the grain. And it doesn't matter if the grain came from a large lesion or a small lesion, you always see it like that. However, if you look at Falciformis spora senegalensis, you also have a black grain, but the cement material is more towards the periphery, and in the middle, you don't see cement material. So it really depends on the species. And the species are also very genetically diverse. So Margella is member of the Sordarealis, Falciformis spora is member of the Pleiosporalis, The bacteria are, of course, genetically also completely different than the fungi. So it looks like that the host is the defining factor, but what is the reason behind it, we don't know.
And what is the difference between the bacterial and the fungal? Do they look the same as well? Pathology.
Yeah, so they both form grains, so that's similar, but the color of the grains is different. So the fungal melanins are usually black and melanized, while the bacterial ones are not. And of course, in the bacterial grains, you see bacterial filaments, and in the fungal ones, you see the hyphae.
Yes, it's
But the immune reactions surrounding the grains, that's similar.
It's interesting, the both. Can I ask Sande a question, Christina? Is that okay?
Yeah, go right ahead.
So, I was very interested in this very promiscuous domain, this WD40, which is also a lubricating agent.
Right.
Yeah. So this WD40 domain, you find it in like some 90-something proteins. It seems to have in multiple copies. So the reason for the multiple copies, do you know? Do you have any idea of what structural components it contributes to the proteins that it's in? Does it extend domains apart for a specific reason? Does it allow domains to fold the specific conformations? Do you have any idea about that?
You need multiple copies of which WD40 proteins you're talking about?
Well, in general, the WD40
In general.
proteins. Yeah, in general. Do you know anything about what structural components?
Yeah, it's very interesting because it's actually in the WD40 repeats. I mean, the WD40, the proteins have the multiple variable number of the WD40 repeat. Sometimes they have a 6, sometimes they have a 20, and then depending on the number of the WD40 repeat, and so their structural scaffold is different. And then also, when I read the review papers about the WD40 protein as a potential drug target, is actually the only conserved motif in the WD40 repeat is the tryptophan aspartic acid that the C-terminal regions. The other region has a very different amino acid flexibility. So even though it looks like they have a better propeller structures, but depending on the number of the repeat, and then also depending on the amino acid, the kinds of the amino acid, their structure is very different. So they mainly play a role as a scaffold of proteins, and then depending on the subtle structural difference and the different interaction partners can bind it to it. So they kind of they have a depending on the different client proteins, so they play some different roles. And then I don't know why they have so many copies of this WD40 repeat evolutionary in the eukaryotic cell, but probably I know maybe the WD40 domain plays such an excellent role as a scaffolding, so providing the kind of the place for interacting or let other protein communicating with each other. So,
but
you
say structural fold, but that last story that you gave, I forget the name of the protein, suggested that it was actually functional as well, right? Because you made those mutations to show that it was.
That the high CO WD40 containing cyclophilin protein. Yeah.
So
you're
that
talking
has a
about
functional role as well.
Yeah.
Irene Garcia, you've raised your hand to ask a question. Could you pop your question in the question and answer box, please?
Ask one follow-up, Sun, on the WD-40s. You mentioned that they have a peptide binding pocket that then can bind also inhibitors. Is that a part of the fold, or is that a different part of the protein? Like, what gives that specificity to different proteins versus, is that a component of just the fold?
You talk about, wait.
In your talk, you mentioned that there's a,
yeah,
you were talking about the fold.
Yeah. You mean this one, right?
Yeah, it was somewhere in here. Yeah. Yes.
Right.
Yes, it's in the context of thinking about drugging them. Yes.
Right. So I got this picture from this very beautiful review papers published in the Nature Review Drug Discovery. So all these reviews are highlighted that this WD-40 protein actually sometimes when there's a protein-protein interactions, then usually sometimes the two protein has a very flat interface between the protein. In that case, is that those interface cannot be targeted for the drug development very efficiently. However, in the case of WD-40 proteins, they have a very group that like a center pocket size, which is, so they have some group like that, and the interaction, the partner protein can interact with it. So in these cases, that there's two small chemicals, and actually they bind to the center of it. So when the small molecule bind to the center of this pocket, so they probably inhibit the not just enzymatic activity per se, and then they actually inhibit the protein-protein interaction between these WD-40 proteins and other partner.
Sorry, so this is a specific interaction because the domain,
yes,
full, and the amino acids could be different between the different proteins that you're showing here.
Between these proteins, so yeah, they have some quite a specificity, I think. Yeah.
All right. Thank you.
So, yeah.
Okay, and I have a question for Wendy about what is the mycetoma material made of? Is it known what the and do you have any idea whether the difference in lesion severity is related to inoculum? Although the person Julia Coe who posed that has pointed out that that's probably very impossible to determine. So what is the cement material made of?
So the cement material, we were not able to characterize it completely. Simply, I don't have the expertise to do that. But what we know is that chitin is part of it, proteins are part of it, DNA is part of it. So we could use various dyes, but we couldn't do NMR because we didn't have that available here. So if anyone can help us there, please do. With respect to the inoculum size, so we needed to inject 4 milligram of Margella mystomatis hyphae in a 500 milligram larvae to get a lethal dose. But if we inject less, we still get mycetoma grains, but the infection is not lethal to the larvae. So then it's still a very good model, but it made it a little bit more difficult for a readout. So certainly the inoculum size does matter in terms in the Galeria if the disease is lethal, yes or no. But for grain development itself, it also develops grains on a very low concentration, so 100 times less. You see grains developing over time.
Okay, thank you. I think there's a last question, Christina,
Yes.
from Michael.
So there's one last question from Mikhail Oselski. Did you have a chance to test the highly specific inhibitors for WD-40 in animal models with crypto? I'm not sure.
Not yet. Yeah, yeah, so I'm thinking about it. For example, like this, the WDR5 is their ortholog is the one of the component of the compost complex. So probably those WDR5 inhibitors may be working for treating Cryptococcus. So that's why we ordered that inhibitors from some company, and so probably we can test them. So I'm not sure, but whether that structure is similar or not, but definitely. So our goal is that we try to make the very easy high throughput screening system after we finish all this work, and then we can identify some critical WD-40 protein, pathobiologically critical WD-40 proteins, and then we can set up some drug screening system for testing drugs or any kind of chemicals for inhibiting those interaction between WD-40 proteins and interaction partners, and then probably we can identify the new drugs. But yeah, but not yet. We haven't tested yet, but I'm thinking about it.
So there's one last comment here for you, Wendy, from Julia, who said you should maybe look at the Canada bioforms, because it sounds like it might be very similar to Canada bioforms, and if there's compounds that are able to combat that, it would be something to look at.
Yeah, thank you. We have looked at biofilms as well because it's our gut feeling that the mycetoma grain is simply a biofilm.
Makes sense. Thank you, both of you.
All
Wanna
right.
close, Christina? Yeah.
Yeah, I think we're going to close there. I wanted to thank both of our speakers, Sun and Wendy, for their excellent talks, and also Gordon and the MRC CMM staff behind the scenes for all of their help in running MycoTalks. We look forward to seeing you in a month's time for the next session, and when we'll have to the last session, the last speakers for this session. Goodbye, everyone.
All right.
Bye, everyone.
Thank
Thank you, Wendy.
Thanks for the invitation. Yeah.
Bye-bye.
Bye, Christina.